Now, in the last video, we talked about the mechanism of an action potential when it moves down an axon. But what happens when it arrives at the axon terminal? So right here, the axon terminal, the axon was back here. We've moved all the way down to the end of the axon. Now we're at the axon terminal. And so at the terminal, we have these vesicles containing our neurotransmitters. And these are essentially chemicals that are responsible for inter-neuron cell communication. So these are really the crux of how our nervous tissue communicates with each other and sends different signals throughout the body. And so we have these vesicles of neurotransmitters here, and we're trying to get these out into this synaptic cleft, which is just empty space between a neuron sending the message and the neuron receiving the message. And so in order to get these neurotransmitters out, we have calcium ions on the outside of the cell. And so when your action potential arrives at the axon terminal, remember the action potential causes a depolarization of the cell membrane. And so when depolarization happens, these calcium ions flow into the cell. And the effect of this is it causes these vesicles to fuse with the cell membrane. So these vesicles are going to fuse with the cell membrane, and the effect is going to cause these neurotransmitters to flow out into the synaptic cleft. So we're going to have these neurotransmitters out here, and it'll diffuse across the cleft until it reaches the receptors on this dendrite right here of the receiving axon. And then the receptors will receive the vesicles and the neurotransmitters, and then that will go on to cause another action potential on this receiving dendrite. And there are a few different types of neurotransmitters. I mean, there's acetylcholine, which is responsible for several functions. There's dopamine, there's serotonin. Serotonin's another good one, etc. And so, yeah, that's basically how two cells communicate with one another. And now, with regard to how we get rid of, well, how do you moderate neurotransmitters, right? So you don't want two neurons to continuously signal each other. There are very few cases in which that's beneficial. And so in order to get these neurotransmitters out, there needs to be a way to sort of regulate when neurotransmitters are in the cleft, because you don't want them there all the time, or at least you very rarely don't want them there all the time. And so in the case of acetylcholine, we have this enzyme called acetylcholine esterase, which actually breaks down acetylcholine and removes it from the synaptic cleft that way. With serotonin, we actually have reuptake inhibitors, which in effect, say this molecule here is serotonin. We have these reuptake inhibitors, which actually take serotonin and bring it back into the presynaptic neuron. And so the effect is reuptake into the presynaptic neuron. And there's one final way, which is probably the most simplest of all, and it's just simple diffusion out of the synaptic cleft. So these just diffuse out of the synaptic cleft and just away from the neurons. And gaseous neurotransmitters like nitric oxide do that.